हेलो गाइस, आई एम शुभम फ्रॉम शुभ ट्यूटोरियल। हम डीबीटी के PYQs इस वीडियो से स्टार्ट कर रहे हैं। तो व्हाट इज द मोटो बेसिकली, हाउ इज द लेवल ऑफ क्वेश्चन, कि क्वेश्चन का लेवल कैसा है और आप इजीली कैसे सॉल्व कर सकते हो, दैट यू विल अंडरस्टैंड। So before we start, subscribe to the channel and press the bell icon to get notified whenever new video is out. First question is, which of these organelles sets are generally equipped with their own genome? So basically this is a 2023 questions, right, of the previous year. इतना सिंपल क्वेश्चन है, ऑर्गेनल सेट्स हैविंग देयर ओन जेनोम। ओके, तो हमें पता है न्यूक्लियस होता है, एंडोप्लाज्मिक रेटिकुलम, न्यूक्लियस गोल्गी, नो, माइटोकॉन्ड्रिया, न्यूक्लियस सेंट्रियोल, नो, सेंट्रियोल में नहीं होता, ER गोल्गी में नहीं होता, न्यूक्लियस तो होना चाहिए, माइटोकॉन्ड्रिया भी होना चाहिए, सेंट्रियोल नहीं, न्यूक्लियस, माइटोकॉन्ड्रिया, क्लोरोप्लास्ट, यस, ऑप्शन नंबर 3, सेंट्रियोल, न्यूक्लियस, पेरोक्सिसोम। सेंट्रियोल पेरोक्सिसोम में तो नहीं होता, राइट। न्यूक्लियस हैव इट्स ओन genome as well as mitochondria, chloroplast. Option number 3. Now the next question, which of the following molecules are involved in the formation of ATP by substrate level phosphorylation during glycolysis? मतलब डायरेक्ट क्वेश्चन आते हैं, ओके। अगर आपको ग्लाइकोलाइसिस पता है देन यू विल सॉल्व दिस क्वेश्चन इजीली। फॉर्मेशन ऑफ ATP, right, formation of ATP by substrate level phosphorylation, and which of the following molecules are involved? तो कौन से molecules involve होते हैं for the ATP synthesis, right? So we know there are two phases. Okay, so ATP formation ये बाद में होता है, right? So the molecules those are involved, that is 1,3-bisphosphoglycerate and phosphoenol pyruvate, glucose 6-phosphate and fructose 6-bisphosphate। ये तो फर्स्ट फेज में है, राइट। नेक्स्ट फेज में होता है, दैट इज A and C, ऑप्शन नंबर 1। ओके, सी दिस इज अ डायग्राम ऑफ ग्लाइकोलाइसिस, कैन सी हियर। तो यहाँ पे ATP सिंथेसिस हो रहा है एंड यहाँ पे, तो हियर इज दिस इज 1,3-bisphosphoglycerate, राइट, and here it is phosphoenol pyruvate. So simple question it was. Okay, next question, match the following is also very easy, बहुत easy होता है match the following solve करना। Now how, see this is condensing, option number B condensing, we know condensing condensation करेगा, so B will be help in chromosome condensation, right, B। तो B4 3 में है, 4 में है, 1 को एलिमिनेट, B4 कॉमन है, C1 कॉमन है, तो C का 1 होगा। ओके, लिंक सिस्टर क्रोमैटिड्स टुगेदर इमीडिएटली आफ्टर एप्लीकेशन, राइट, तो दिस गॉट कंफर्म। अभी A एंड D का हमें देखना है, वेरी इजी। जो यूक्रोमैटिन होता है दैट इज बेसिकली एक्टिव जीन्स, ओके, एक्टिव जीन्स, so this will be 2, A2, partially decondensed region of chromatin with active genes in region, right? So A2, that is an option number 4, and D will be D, highly condensed region of chromatin with inactive genes, right? यूक्रोमैटिन एक्टिव जीन्स होते हैं, एक्टिव, and heterochromatin is inactive. How to remember this? I will just tell you. We know eukaryotes and prokaryotes. Okay, prokaryotes में न्यूक्लियस इज प्रिमिटिव, न्यूक्लियस है, not well developed. In eukaryotes, mean nucleus is developed. Okay, or we can say means the structure is organized. Membrane hai. So when we say U, we can also say true nucleus. Like this, when U word comes, it is basically true thing. तो जीन्स है लेकिन एक्टिव जीन्स, यू कैन रिलेट लाइक दैट. तो डेवलप स्ट्रक्चर, यूकैरियोट्स डेवलप होते हैं देन प्रोकैरियोट्स. तो यू इज बेसिकली एक्टिव, यू क्रोमैटिन इज एक्टिव जीन्स, यू क्रो. I hope you understand this. Next, EcoR1 recognizes the sequence 3AATTC, a stretch of linear DNA with 6 DAATTC sites. Upon digestion with EcoR1 will give rise to 8 fragments, 7 fragments, 6 fragments, 5 fragments. Very easy. See, linear DNA है. Suppose this is having one site, cleavage site. Okay, एक EcoR1 साइट है, तो ये एक फ्रैगमेंट होगा, ये एक फ्रैगमेंट होगा. ओके, इफ सिंगल रेस्ट्रिक्शन साइट है तो देयर आर 2 फ्रैगमेंट्स. ओके, so when suppose I say there are 2 restriction sites, one, so this will be for 1, 2, 3, 3 fragments. अगर 2 sites है तो 3 fragments. ऐसे अगर 6 से sites है, 6 sites. So what you want to do in linear DNA, case of linear DNA, 6 plus 1, 7. एक फ्रैगमेंट को ऐड देना है बस. So the option answer will be 2, 7 fragments. Okay. One more thing, अभी linear DNA का तो हमने देखा. Just add 1. If there is a circular DNA having single restriction site, so this will be equal to the number, right? Means यहाँ पे कट हुआ, then it will be converted into linear DNA. If there are 2 sites, there will be 2 fragments, 1 and 2. Okay, so it will be equal to the number of sites. Means here there is only one site, so fragment will be 1. If there are 2 sites, fragment will be 2. If there are 6 sites, fragment will be 6 only. Right? Let's see. This is 1, 2, 3, 4, 5. 1, 2, 3, 4, 5 and right? Equal fragment we will. Let's erase this. Make place for the next question. Lichen is an example of symbiotic relationship between. Direct question है. If you know this symbiotic relationship, it is between the algae and fungi. Option number 3. Next question. Electrons from excited chlorophyll molecule of photosystem 2 are accepted first by. Okay, photosynthesis अगर पता हो जो pathway है, if you have studied, you will know this. Okay, so this is again an important concept, right, of plant photosynthesis. तो बेसिकली फोटो सिस्टम 2 से इलेक्ट्रॉन्स कौन एक्सेप्ट करता है फर्स्ट? दैट इज प्लास्टोक्विनोन, ऑप्शन नंबर 4. Okay. Okay, this is the pathway to show you. I will recommend that make a screenshot. This is very important for every exam, right? So this is photo system 2. Okay, जब लाइट आई तो एक्साइट होगा and this will go to PQA, PQB. These are plastoquinones, right? This is the pathway. Also, I showed this glycolysis. This is also important for every exam. Make a screenshot. I will erase this. So you can make a screenshot. This is very nice and important pathway. Okay. Which of the following enzyme is not a part of DNA replication machinery? Okay, DNA replication में कौन सा एंजाइम इन्वॉल्व नहीं होता? So we know DNA helicase is involved, primase is involved, DNA polymerase is involved. So the answer will be DNA endonuclease, right? जो involved नहीं होता. Okay, we have to see carefully. This not, right? Not involved. These are direct questions. As हमने molecular biology देखा है पूरा, the videos are available in the playlist, right? The common metabolic precursor of amino acids such as proline and glutamine is. Okay, common metabolic precursor for proline and glutamine is 4 alpha ketoglutarate. Okay, see there is one diagram here again. You can see here. इसका भी स्क्रीनशॉट निकाल सकते हो आप लोग. First, again, this is important. Common metabolic pathways. So here it is alpha ketoglutarate. Here are the amino acids: arginine, glutamate, glutamine, histidine, and proline. Acid rain is caused by carbon dioxide, hydrogen, biomethane, nitrogen dioxide. Nitrogen dioxide. Acid rain is caused by nitrogen dioxide. Which of the following methods cannot be used for ab initio determination of 3-dimensional structure of a protein? Okay, 3D structure के लिए हम क्या नहीं यूज कर सकते? Which of the techniques? X-ray crystallography 3D structure, yes. Nuclear NMR, yes. Cryo-electron microscopy, yes, we can use. Conventional mass spectrometry, no, this is not used for 3D, right? So option 4 will be the answer. Okay. So these were the questions for today. That's it for this video. I hope this video helps you. If you find this video helpful, do let us know in the comment section. Like this video, share this video with your friends. Subscribe to the channel. Stay tuned for the upcoming video. Thank you.